All right, guys, thanks for coming today to today's seminar about multiomics and methylation. I'm very grateful to Catherine for putting this together as well, and to SeekGene for coming again in person to run a really cool demo with us. We've had really good experience with both Chameleon and SeekGene so far. Like with Catherine, we've worked on PipSeq in the early days, worked on Curio Tracker, and we've tried all that out with great success. And the latest addition to our portfolio is SeekGene, where we have last year trialed their spatial products. And there's a really exciting pipeline that Unica will tell you about after I finish my blurb, revolving around spatial methylation multiomics, which is really exciting as well. But today we wanted to talk and focus a bit more on single-cell methylation, not spatialized context, but just general single-cell methylation multiomics. And what we're doing this week is trialing some reactions on some interesting samples that come from in-house, but also from experts in the field, such as Heather Lee, who was giving a short talk afterwards, who is kind of leading that single-cell methylation field in Australia right now. And then we have a talk by Ruth Pitsley as well on what's happening in-house. And there's a lot more interest in-house beyond that as well. So I'm really excited that we have the opportunity to test this product at Garvan Genomics. If you don't know much about Garvan Genomics yet, we've got a poster standing up here. So we're basically a one-stop shop for all things single-cell and spatial. You can literally drop off your blocks, or you can drop off your cryo vials with us. We do all the thawing, we do the isolation, we can sort the cells. We can also ship tissue interstate, if that's interesting. And then do basically everything in-house, including sequencing, with a turnaround time of the current record being 8 days from receiving a sample to returning data to customers. So we're reliable, fast, efficient, and we also interface really well with the internal Garvan platforms and services. So if you need a true one-stop shop solution, like, come to us and we can mediate everything in your entire project. Yeah, without further ado, I wanted to introduce Unica and the SeekGene team to tell us all about this really cool methylation product.
Thank you, Chris. Hello, everyone. We're really happy to be here with you. Today, I'm bringing our new technology, DNA methylation multiomics. And this demo in Garvan is actually our first demo aboard. Yeah, that's true. Because this is very new product, just launched in February of this year. So I'm very excited to be here and bring this technology to show you how SeekGene is doing for the single-cell field. So to start, I want to just briefly introduce what we have for the current challenges for DNA methylation techniques. So firstly, the conventional DNA methylation techniques may have limited throughput. So basically, like plate-based techniques, we can only run like 100 cells or to basically 1,000 cells. And another thing is it's bulk sequencing. It's not single-cell resolution. And the matrix are highly sparse. It's difficult to identify rare cell types, for example. And the other method we use is mostly like biosulfate conversion. So this might have some DNA damage to our cells. And the last thing is MultiOmics only. For example, the techniques from ScaleBio. So they can only like detect the DNA methylation information, and the RNA information is actually missing. So based on that, I'm gonna show you some timeline and briefly introduce other current methods for single-cell DNA methylation sequencing. For example, the plate-based, and also, for example, here for the cabinet and some MET. So as you can see on the below table here, different methods. Some are low throughput, some are medium throughput, some can capture different number of cells, some from CT conversion using biosulfate, some also using enzymes. So the library prep may also be different between different technologies. But what I'm given today, which is our technique launched in 2026 this year, is SeekGene single-cell Methylation+scRNA multiomics. These ones we can capture 5,000 cells to 12,000 cells, and it is enzymatic conversion, and also multiomics that we can get both RNA information and DNA methylation information inside the same cell. So I also made this kind of figure to show you how the location and position of different technologies are different in terms of throughput, multiomics, and you can see on this table here. And the position of our technologies in this, as you can see, high throughput and multiomics. So just to highlight some advantage of our technologies here. Firstly, is multiomics with single cell resolution. It's high throughput, it's genome-wide coverage, and easy to use with a friendly workflow, as we may see in the demo experiment. So let's get started. As you can see, this slide is the basic principle of this DNA methylation multiomics kit. So what we start is you can have both cells or nuclei. It depends on what you want to see. The cells might be preferred because we have the cytoplasm RNA information, and the nuclei may be more preferable for like brain cells. So you'll do like irregular shape cells will be more suitable for nuclear isolation. And then we'll do a fixation with methanol and the nucleosome depletion. And after that will be TM5 tegmentation using TM5 enzyme. But what's different thing about this TM5 enzyme is very different from the TM5 we used in ATAC experiment. This TM5 enzyme is specially designed with a linker and a biotin incorporation. So after that will be the droplet system with our droplet machine, the instrument to do single cell separation and labeling stuff. So followed by the reverse transcription and then the B separation to separate the DNA and the RNA information. So finally, we will generate two libraries. One is RNA library, cDNA library. The other one is DNA methylation library. And here is a design of our barcode beads. We're using the same barcode beads to capture both RNA and DNA tegmented fragments. So this one is cooperated with the TSO sequence. It's very similar to what we used for the single-cell 5' RNA-seq. So just to show you how they are captured. For the DNA fragments, we will add a linker, like a bridge, to link the TSO sequence with the ME sequence from the TM5 sequences. And the mRNA is very similar to the regular 5' library. So here is a library structure. The upper one is transcriptome library, the below one is methylation library. As you can see, we have a sequence for linker and ME, and here is very similar to the 5' library. What's interesting about this technology is that we can get multiple profiling from the same cell. One is epigenetics, one is genomics with the CNV analysis, transcriptomics with the mRNA, and immunology possibility with the VDJ or B-cell T-cell sequencing. As we showed in the last slide, we are using TSO sequence to capture mRNA. So that means we can have a potential to incorporate with the VDJ sequencing as well. So you just do additional amplification using T-cell, B-cell probes, then we can get both of the information from the same cell. Regarding the sequencing platforms, we are compatible with Illumina or GenMa and using P150 sequencing modes. And the recommended sequencing depth will be, for the methylation, will be 12.2 gigabase per cell or per nucleus. And that will be 350 to 700 carries per cell. For the RNA library, it's very similar to the regular one, 100 gigabits total or 25K to 50K reads per cell. So it might also be interesting the sample requirements. For the DNA methylation, we can do both fresh cell, fresh tissue, or frozen cell, frozen tissue. And we have some requirements in terms of the quality of our cell suspension or nucleus suspension. For example, the viability greater than 80% for the fresh cells, and the aggregation rate, nucleus integrity, something like that. And for the tissue, for example, fresh tissue or frozen tissue will recommend a volume of greater than 3,000 milligram. And this is some sample types we have done so far. We have some experience with this kind of tissue types, what we have done in the, for our customers in China. We have also done some statistics of the data we have run so far. So just showing you, for example, PBMC sample, colon cancer, some other cancer types like lung cancer, and also prostate cancer, embryo, some different tissue types we have done so far. And we have found that the average median genes is greater than 1,000, and the average number of CPGs is greater than 50,000. And more data performance metrics is here. So as you can see, we have done like from humans, from mouse, different tissue types, PBMC, cancer, brain, embryo, and other tissue types as well. And the median genes is in this column here, and this is the estimate number of cells, and here is the number of CPG of median cell. So just for you to have a reference. For the basic cell clustering, I've chosen 3 examples here. So these ones are from human prostate tumor with whole cells. So it's cell suspension. And the middle one is from mouse brain. We have done nuclear isolation. So this is from nuclear suspension. And this on the right-hand side is mouse liver. It's also whole cells. So you can see that we can obtain very clear cell clustering. Every clustering is identified and also distinguishable. For the final data, I've given 2 examples here. One is from prostate tumor, one is from breast tumor. They're all both from humans. And which I want to show you here is with different sequencing depths, they may have different like CPG number of median cell and median genes per cell. And also this is number of cells being captured. To compare with existing current methods of single-cell DNA methylation multiomics, this method is called cabinet. It's also a single-cell level multiomics with DNA methylation. So what SeekGene stands here is that at a similar read depths, we can still achieve higher genome coverage and with lower saturation compared to other techniques. So finally, about the case study. This is a real case study from one of our customers in China who was studying aging. And so they have chosen different PBMC samples from different age groups and different gender. So what we can get here is figure 1 here is RNA-based clustering, and figure 2 on the left-hand side is methylation-based clustering annotated by mapping RNA labels. And this is the methylation level mapped to the cell clusters. As you can see that T cells, B cells, and monocytes and NK cells are all present in those PBMC samples. So just to note here is RNA is short-term adaptation and DNA methylation is more like long-term epigenetic memory. So we want to find some linkage between RNA and DNA methylation. Interesting is that when we zoom into the subpopulation of different cell types, we can find that the naive immune cells specifically, they show very global higher DNA methylation levels compared to other cell types. And the other point we found is the family gene drive, the drive gene from PCDHGA1. This family gene drives very immune cell aging at the epigenetic level. So this is also consistent with the publication, with a reference from one of the paper. So we also have compared our epigenetic pattern with another paper from the publication here. And for example, the A column here is the Atlas project data using our technology. And the B column here is the cells with methylation level. C is the cells with coverage of detected methylation sites. And then finally, the column D is the published data, published paper data. And you can see that column A and column D, they are very consistent. So that's also to validate our technology with accuracy and reliability. Another case study is about cancer and oncology. You may know that epigenetic is also closely related with cancer study and cancer development. So to address some key oncology questions, for example, clonal evolution, immune interaction, immune response, what's our methylation technology can bring or investigate is that the methylation plus CNV analysis or RNA profiling, VDJ profiling. So this is from a breast cancer cell line. On the right-hand side, this is a methylation pattern, the CNV analysis pattern, and to compare it with the paper data using WGS CNV profile, and we found the data pretty consistent and with high coordinates. Another case study is about prostate cancer. We have done a very large atlas of over 100 samples from prostate cancer tumors. So this is just some part of the data from that atlas study. For example, this is from the table from RNA library, and here is from the DNA methylation library. When we're doing the CNV analysis, we actually have two different analysis methods for these ones. One is copycat based on the RNA information. One is copykit based on DNA methylation information, and they are quite giving us different results, which you can show here. This based on the RNA data, we only detected 2 clonals. But with the DNA methylation data, we actually identified 5 different clones in the epithelial cells. And that means using DNA methylation data will generate more clones, and different clones will show distinct clustering in the UMAP plot. Just some background information here. What's different from copycat, copykit, and inverse CNV? So copycat and inverse CNV, they are based on single-cell RNA-seq data, and copykit is based on single-cell DNA sequencing data. And all the RNA-based CNV tools have some limitations because the gene expression is not purely identified by the copy number, and they may have some influence by cell cycle, stress, inflammation. These factors can all interfere with our CNV signals. So that means the copykit using DNA methylation data is more accurate than RNA-based methods. And what we get from the results is further validating this principle. So you can see that this slide is showing the adjacent normal prostate tissue results. For example, the smooth muscle cells and the fibroblasts, they are genomically stable. They are normal cells. They are not tumor cells. So these cells shouldn't have any CMVs. But when we look at the results using RNA-derived CMV, like copycat, it gives us some false positives, for example, which is showing here. But when we look at based on the copy-kit DNA methylation data, so these ones are not detected with the CMV. That means it's more accurate. On the other hand, we also looking at the prostate cancer cells like epithelial cells, which is showing this cluster, and compared the difference between copycat and copy-kit is that we can have a clear pattern of CMV in the DNA methylation derived data compared to the RNA derived data. So finally, to highlight the SeekGene DNA methylation multiomics, one is that we are getting multiple profiling from the stem cell methylation, RNA, CMV, and VDJ. We can do high throughput profiling up to 12,000 cells per chip. And our method is based on enzymatic conversion of C to T. This is more user-friendly to our cells. And the other point is that we are doing native data alignment. You don't need to do separate experiment for the methylation and the RNA sequencing. Final slides about some frequently asked questions. One is that, can we distinguish HMC and MC detection? But unfortunately, current methods, we cannot distinguish between HMC and MC, but we are cooperating with the research group specializing in the DNA methylation. We are cooperating together to explore methods for differentiating these two methylation in the future developments. And the second question is most frequently asked, can we do multiplexing? So the answer is, if you do same SNPs, like genotyping-based demultiplexing method are still compatible with this technology. So just like I showed during the aging studies, they are getting PBMC from different age groups and different gender. So they can kind of demultiplexing by the Y chromosome alignment. But multiplexing methods like hashtags are not currently compatible with this kit. So third question is about genome coverage. I'm sure some of you already see some data from us. Some customers said like the genome coverage per cell is pretty low, like only 0.3% for the sample. But what I would like to say here is that for the total library genome coverage is still generally larger than 92%. And at around 1 TP sequencing, the median cell coverage is typically between 0.2 to 0.5%. And at a 2 gigabase per cell sequencing depth, we can get like 1.5 to 3 million CpG detected per cell, and the total CpG detected overall will be 28 millions. Just show you another example of PBMC. This is from very earliest internal testing, and this one we are doing sequencing of 2.7 TB sequencing data with around 4,000 K-reads per cell. And with this one, we can get a higher genome coverage rate of median cell of nearly 4%. But just compared with the low throughput platforms, they are like doing usually lower than 2,000 cells. So they can get more sequencing depth for each cell. That means that the gene coverage per the genome median cell for the low throughput platforms will be higher than the high throughput platforms like SeekGene. So what we recommend is 0.2 gigabase per nucleus and up to 12,000 nuclei or cells per experiment. So there is always a trade-off between throughput and genome coverage per cell. But our high throughput platforms, the advantage will be more for cellular heterogeneity analysis, like epigenetic landscape mapping and cell-specific methylation level or population level comparisons. Yeah, that's all I want to share for today. Thank you very much for watching.
Do you have any questions while we're waiting for Orion to...
Yeah.
Do you have any questions?
Is your conversion, is your deamination, is it C to U or is it methyl C to DHU? Which one is it?
We are using the NEB kit from Bell, US, so it should be...
From you, oh, the NEB kit, so it's C to U.
No other questions? So I'm going to leave in Orion's people hands. He's going to talk us through. They've done a bit of a couple of tests on some of the other products that we've SeekGene. Orion's going to go through some of his evaluation as to those things as well.
Let me disable my red filter so it's a bit easier to see. But yeah, we at the sort of platform have been interested in exploring a couple of different types of single cell and spatial methods that are maybe a little bit sort of up and coming, shall we say. And so this has been the reason behind our collaboration with SeekGene to really explore a lot of novel workflows for, as we say here, undertapped single cell and spatial biology. So I'll whiz through these slides. Everyone's pretty much seen and heard of single cell at this point. Poly-A capture, very standard. You can kind of think of this as a very in-depth way of being able to look at one particular sample, let's say. And then, of course, you've got spatial transcriptomics, where we're looking more at a sort of neighborhood type resolution. So you might think of this as kind of how something is constructed, let's say. And then if we think back to where a lot of these technologies actually started, it sort of started off in bulk methodologies. And bulk transcriptomics and sequencing can sometimes get a little bit of flack as not being sort of high resolution or providing the level of information that we're sort of accustomed to with single cell and spatial methods. But actually, there's a sort of huge amount that we can take away from bulk methodologies because of their complexity and ability to be easily manipulated. And so when we're talking about things like transcriptomics, we're talking about the split between looking at mRNA and total RNA species, as well as other small RNAs. We're talking about DNA. A lot of these technologies have not yet been translated to single cell and spatial techniques. Epigenetics, translatomics, and protein analysis, also very similar here. So our goal in the platform is to sort of help to enable this transition from a lot of these bulk technologies to the sort of single cell and spatial resolutions in a technologically meaningful way. So just starting off by talking about the transcriptomics side, again, we've put together a sort of a bit of a comparison across different types of single cell technologies that we've tested in the platform on either a model cell line or PBMCs. And again, I won't go through all of the detail. If you've got any questions, feel free to talk to any members of the genomics team. But you can see that there's sort of a whole gamut of different types of variants that we have here. So things like the capture method, the chemistry, the number of cells, the sort of strengths and weaknesses of different approaches. And what I really wanted to highlight was that sort of chemistry column here. So you've got a lot of focus on poly-A methods. So these are your conventional 10x, 3' and 5'. You also have the sort of emergence of additional types of chemistry, like random primers, as well as a combination of poly-A and random primers from other workflows. So really the reason why we're starting to see a bit of an exploration into new types of chemistry is that poly-DT capture, so your sort of poly-A methods, really only get you, a fairly important part of the transcriptome, but a part of the transcriptome. And so when we're thinking about understanding other types of RNA diversity, that's where we really need to be looking at other methodologies. So again, that focus on mRNA kind of potentially obfuscates things like long non-coding RNA and other regulatory RNAs that might be interesting. And secondly, the sort of focus on the 5' or the 3' end of the gene really means that you're not going to be able to capture that sort of true diversity that you find across the whole gene. So firstly, we've kind of assessed a couple of these different technologies on PBMCs or a mouse osteosarcoma cell line. Again, I'll whiz through some of this data, but we've examined things like sequencing saturation, the number of UMIs per cell when you downsample to make the comparisons a bit more even. We've also got information about things like VDJ recovery for your 5' methods as well. And so again, both in terms of the number of TCRs you recover, as well as the productivity of those TCRs. And the one I wanted to highlight a little bit more was this element of being able to capture long non-coding RNA. So long non-coding RNA are starting to be more and more appreciated. They often do have poly-A tails, but a large proportion of them also lack poly-A tails. So you're only going to get an incomplete sort of picture of long non-coding RNA if you stick with your traditional methods. So here we tested and looked at the levels of long non-coding RNA in the same osteosarcoma cell line across a lot of these different types of technology. And the sort of SeekGene fast-seq approach, which uses random primers, ended up giving us the greatest percentage of long non-coding RNA in our data. And so what was quite interesting was when we clustered our osteosarcoma cell line based on all genes, you get the UMAP on the left, and if you take just the long non-coding RNA in that same data set and do a similar clustering, you end up getting a similarity index of about 0.56. So what this means is that about 50% of the information is conserved across sort of the full poly-A transcriptome, as well as the long non-coding RNAs, but you do get a divergence in what kind of cells and clusters are called just on the long non-coding RNA information. The last thing I just very quickly highlighted is that different types of technologies have different biases towards parts of the gene body. And here, again, based on our own in-house data, we've actually compared this across some of these more emerging types of workflows, which are trying to look at more of an even gene body coverage. So again, we see much better even representation across the middle of the gene body using methods that involve random primers. So again, if that's of interest in looking at isoforms, that's certainly something worth considering. The next thing I'm just going to very quickly talk about is on the spatial side. So when we sort of refer to spatial or this sort of evolution of spatial like biology, etc., a lot of the times we are just focusing on transcriptomics because it is such a fantastic layer of information, but it's just one layer of information regardless. So here we've kind of been interested in the best methods for transcriptomics on the spatial side. There are kind of maybe 3 methods that we're sort of looking at, or at least currently sort of actively investigating. So one is the sort of seq-space method, which I'll talk about in a little bit, that has unique applications in some experimental settings. Then you've got your sort of classic Visium HD, where you're capturing RNA in pixels. And then you've got your sort of Xenium or Atera type applications, where you've got an actual in situ imager there as well. So the sort of 2 applications on the right are probably more familiar, but the reason why we were quite interested in SeekGene is because it offers quite clean and single cell resolution levels of spatial analysis. So rather than the diffusion of transcripts or the capture of transcripts, instead nuclei are spatially barcoded. And then afterwards, those nuclei can be put through your favorite single cell assay. So you end up getting spatially resolved single nuclei RNA sequencing data. And this has particular use cases when you're interested in segmentation artifacts and overcoming those, as well as minimizing the effects of transcript diffusion. So we've run the seq-space assay on some mouse brains, and I'm not going to go through all of the clustering, but I think what was quite standout to us was the fact that you could see all of the different cortical neuron layers that emerge quite cleanly in the cortex. The other thing to mention is that this analysis took about 2 to 3 days, whereas your analysis of things like Visium and Xenium can take a lot longer and be more computationally intensive. The last thing I'll mention on this side of things is that because your nuclei are individually separated, you run into much cleaner data. So when we're looking at our preliminary mouse brain data, across the different mouse neuronal clusters and populations, we see the sort of prototypic marker genes basically only being expressed in those particular clusters. When we compare that to something like Xenium Prime with the cell segmentation kit, again, it's quite clean, but there is sort of particular bleed through of astrocyte markers. And it's worth noting that the cell segmentation kit doesn't work particularly well for astrocytes. They're not that well resolved by the 18S ribosomal staining. And so because, again, you're taking a section, the segmentation can be more challenging. And then when you compare that to other technologies like Xenium and Visium, which have slightly sort of lower resolution without the cell segmentation, again, you see more artifacts as well. And the last thing to just mention is that SeekSpace and also Visium HD are both sequencing-based imaging platforms. And what this means is that when we're starting to think about how we can translate them to epigenetics and other types of technologies that require sequencing, these are going to be the ways forward. Although something like the Xenium or Atira approach is probably very well suited for high-resolution spatial transcriptomics. So really, hopefully, what we can get to is a point where we've got single-cell methodologies, spatial methodologies, as well as the sort of full gamut of biology in that DNA, RNA, and protein stack, and we should be able to resolve biology much better. And so we are, as Unica mentioned, testing our single-cell methylation workflow. So apologies, I took this photo this morning. It might not be the most flattering, but here to tell you some more will be Ruth and Heather, and we're really pleased to be working with them. Thank you very much.
While Ruth's getting set up, does anyone have any questions on these other two techniques, SeekSpace or the SeekGene techniques?
That's a good question. Orion, do you know if that you can find transcripts from transposable elements more easily?
Oh, that's a good question. We haven't looked in our data specifically. I would assume that you should be able to because the gene body coverage is a bit more even. The problem might be that you'd probably want to sequence a little bit longer rather than your sort of classic 90 base pairs of sequencing, because that might be a little bit more challenging to then map it. But you certainly should be able to.
Interesting to check.
Is there other questions?
Thank you. Hello, everyone. I'd like to start by thanking SeekGene and Chameleon Science for the invitation to talk today about some of our team's work using bulk DNA methylation approaches and some of the advantages I can see with using single-cell methylation in the future to better answer some of our research questions. So I thought I'd begin by going right back to some of the basics about DNA methylation. As DNA methylation is an epigenetic modification to the DNA that typically occurs at a cytosine guanine dinucleotide, or CpG site. And it's defined as the addition of a methyl group to the 5th carbon position of a cytosine to make 5-methylcytosine by the action of methyltransferases. But DNA methylation and the distribution of CpG sites across the genome is not uniform. So in a normal cell, you have CpG-poor gene bodies and intergenic regions, which tend to be methylated, whereas around the promoters of genes, there are clusters of CpG sites termed CpG islands. And these are often unmethylated when the gene is being actively transcribed and methylated when the gene is silenced. You also have regulatory regions, such as enhancers, which often have reduced levels of DNA methylation, reflecting their activity. And so through this patterning across the genome, DNA methylation helps to regulate the accessibility of the genome and therefore gene expression and cell function. Over the last few decades, DNA methylation studies using bulk sequencing approaches have revealed some major findings. Firstly, that cancer is an epigenetic disease. So cancer used to be thought of as primarily a genetic disease caused by genetic mutations, but bulk methylation sequencing has shown that most cancers have a global loss of DNA methylation associated with genomic instability, and also regions of localized promoter hypermethylation, for example, at silenced tumor suppressor genes. And I can show this data here from some work from our team, where we used bulk whole genome bisulfite sequencing to compare genome-wide methylation at almost 28 million CpG sites across the genome of a prostate cancer cell line compared to a normal prostate epithelial cell line. And so you can clearly see here that there's a considerable loss of methylation in the cancer cell line. The same dataset also showed increased methylation at specific CpG island gene promoters. And here I'm showing a very well-known example, which is hypermethylation of the GST-Pi gene promoter CpG island. And this occurs in 90% of prostate tumors. And you can see clear differences between the tumor cell line and prostate tissue in red compared to the normal cell lines and normal tissues in blue. So returning to these major findings, another popular way of analysing genome-wide DNA methylation from bulk samples is to use bisulfite conversion combined with DNA methylation microarrays. And these first became available in the late noughties, and for the first time they allowed genome-wide profiling of DNA methylation across large sample collections and cohorts. And one of the features that really stood out from this new data type was that DNA methylation was associated with human age, irrespective of tissue or cell type. And this phenomenon is so robust that researchers have now made epigenetic clocks that can predict a person's chronological age from a DNA sample with a surprising degree of accuracy. Bulk methylation studies also showed us that environmental exposures can leave traces on the DNA. And one of the most famous examples of this is DNA methylation of specific genes that are associated with cigarette smoking history. And so these methylation changes are persistent and they can be observed even years after a person has ceased smoking. Of course, DNA methylation also has a long-established role in genomic imprinting, where one parental allele is silenced, and also X chromosome inactivation in females. And finally, bulk studies of pure sorted cell populations have shown that cell types have highly distinct DNA methylation profiles that reflect the silencing of genes that occurs during cellular differentiation. To illustrate this, here I'm showing some work from our team where we use DNA methylation microarrays to profile bulk DNA methylation in a rare type of breast tumor called a Phyllodes tumor, which we analysed together with public methylation data from other breast and soft tissue tumor types. And on the TS&E plot on the left, you can see that the DNA methylation was able to cluster samples by tissue type, which demonstrates its tissue specificity. But researchers have also been able to leverage the fact that individual cell types have distinct methylation signatures to develop bioinformatic tools to deconvolute bulk DNA methylation data such as this. So for our study, we were able to use these tools to estimate the proportion of epithelial, fibroblast, and immune cell types in each sample. And it showed us that the Phyllodes tumors in purple had a much higher fibroblast content than the breast cancers in dark blue, which are predominantly epithelial. And this is exactly as expected, as Phyllodes are a type of fibroepithelial tumor type. So many methylation studies of the past 2 decades have likely been confounded by these cell composition effects. And when we didn't have the tools at the time to deal with these cell composition effects appropriately, we can now use these bioinformatic methylation approaches, but really I think the better thing to do would be to use single-cell approaches in the future to be able to tell what are the specific changes happening in each individual cell type. So in other work, our team has also spent many years studying DNA methylation in prostate fibroblasts. And fibroblasts are a type of stromal cell that produce proteins like collagens to make up the structural scaffold of a tissue supporting other cell types. They're a normal component of healthy prostate tissue, but in cancer they are altered and produce proteins that change the scaffold of the tissue in ways that help the tumour to grow. So in our research, we're interested in understanding whether epigenetic differences between these non-malignant and cancer-associated fibroblasts might be a causative feature of prostate cancer progression. So we've worked closely with the team at Monash University, including Gail Risbridger and Mitchell Lawrence, to study these fibroblast cells. And the fibroblasts we work with are taken from radical prostatectomy samples, from which non-malignant prostate fibroblasts are taken from a benign region of the prostate, and cancer-associated fibroblasts, or CAFs, from around the site of the tumour. And then the pro-tumorigenic function of these CAFs has been confirmed with in vitro and in vivo co-culture techniques. And we've previously performed whole genome bisulfite sequencing on these samples and found over 5,000 consistent DNA methylation differences in the CAFs compared to the MPFs, with many showing associated differences in gene expression. And so here I'm showing an example at the PITX2 gene, where you can clearly see that there's reduced methylation in every CAF compared to their matched MPF. In a subsequent study, we profiled more prostate fibroblasts using DNA methylation microarray methods. And again, we saw highly consistent DNA methylation differences across many patients, providing more evidence that DNA methylation may underpin the pro-tumorigenic phenotype of CAFs. If we display the data, the genome-wide data in an MDS plot, it showed that the CAFs have a very distinct profile away from the MPFs. But what it also showed was that there seems to be more individual heterogeneity between individuals in the CAFs compared to the MPFs, which could perhaps be driven by within sample cellular heterogeneity. So to investigate this further, in more recent work, we've performed single-cell transcriptomic analysis of some of these CAFs. And what we've observed is that there is a high degree of within-sample heterogeneity, identifying many different CAF subtypes. So looking ahead, we want to continue our DNA methylation studies of CAFs, but we want to make use of the greater resolution offered by new single-cell methylation techniques. We're hoping that these methylation approaches will be able to provide information beyond what single-cell transcriptomics can offer alone. So, for example, single-cell methylation analysis will allow us to identify methylation changes that are occurring in specific cell subtypes, as well as allowing us to understand whether our bulk data was dominated by a specific cell type. Also, being able to directly measure the relationship between methylation and expression will aid our understanding of the gene regulation in CAFs over previous methods where we've always had to average our data across bulk populations. And as methylation changes are more stable than gene expression changes, they're less likely to be altered by short-term influences like cell culture stress. So single-cell methylation may allow us to distinguish transient from long-term stable differences in cells. And finally, because methylation reflects cell identity, single-cell approaches may provide a better way of identifying and tracking cell lineage. For example, helping to answer questions about the cellular origins of different CAF subtypes. And so with that, I'd like to say thank you to everyone who contributed to this work.
Thanks, Ruth. Does anyone have any questions? Sorry. Yes?
I have one question with the epigenetic clock types. So those clocks were trained on bulk data. You think that would still be recapitulated at the single cell level?
Yeah, I think it would. Like, they've even found in cells. Yeah, I think that more and more people have tried to purify cell populations and keep these bulk analyses. I think what they find is each cell has its own specific clock. So I think there's more work to do to understand why that's happening and what it means biologically.
Thank you. Just to give you some background as well, I don't know if it's very clear, but we're actually running the demo this week with the SeekGene team, which is why they're here, on with a number of groups doing methylation kits, so using the RNA methylation kits. So watch this space. We hope to be presenting some data maybe later in the year, possibly in conjunction with this as well. I want to introduce Heather Lee, Associate Professor from the University of Newcastle in Genetics.
Thank you. Thank you so much for having me today. It's a pleasure to be here because I love single-cell analysis of DNA methylation. I've been doing it for far too long, actually. So I guess following on from what other people have already presented today, I'm going to highlight a few things that you can do with this kind of technology. Drawing on some of the work from my team in the recent leukemia paper, but also on some other published studies. So first of all, I wanted to show you that single-cell analysis of DNA methylation can introduce treatment-induced epigenetic heterogeneity. So in my team, we've been studying hypermethylating agents, which are therapies used to treat patients with the blood cancer acute myeloid leukemia and also myelodysplastic syndrome, which can precede AML. And these drugs are cytidine analogues that are actually incorporated into DNA during replication. But because of a change in the chemical structure, these drugs can't be methylated. And basically, it leads to the loss of DNA methylation during subsequent cell replication, cell division. There you go. There's the 5' carbon on the cytidine base that is normally the site of methylation, but changed to a nitrogen atom in azacitidine and decitabine, these two HMAs. So for a long time, these drugs were thought to work by allowing re-expression of genes that were normally silenced in cancer, such as tumor suppressor genes, DNA repair genes, and differentiation genes. But more recently, it's become much more accepted that these drugs also work by allowing expression of things that are never normally expressed in somatic cells, such as cancer testis antigens and endogenous retroviral elements. And these drugs are useful therapies because they have relatively mild side effects. And that means that there's a treatment option for older or more frail patients for whom the standard cytotoxic chemotherapy could itself be fatal. So these drugs are used to treat those more vulnerable patients, and some patients have a really durable benefit from these. How do I do a point? Okay, yeah, so some patients have durable benefits from treatment, but other patients don't have any benefit at all from this therapy, and we don't have a way of telling who's going to do well and who's may as well not take the drug. And although we know that they work through loss of methylation, the loss of methylation is really similar between responding patients and non-responding patients. Okay. So we wanted to use the single-cell methods that I developed during my postdoc in Cambridge to try and understand if there were differences in how individual cells responded to these HMAs and whether that could also influence how patients respond to treatment. So the data I'm going to show you from this paper was using SeNMT-seq, which is a plate-based bisulfite assay, where we can obtain information on gene expression with the SMART-seq2 protocol and DNA methylation at CPG sites. And we also added a bacterial enzymatic conversion to label open chromatin with GPC methylation. So then afterwards, you can separate that and get information on chromatin accessibility. So these are good for getting high-resolution multiomic information from one single cell. We get information on DNA methylation across the whole genome. And we can also record information when we sort the cells into the plate using index FACS analysis. But this method has really high sequencing demand, it's low throughput, and there's complex library prep and data analysis. So first of all, we treated these AML cell lines with these HMA agents, azacitidine in purple and decitabine in aqua. And we saw that most of the cells that were treated lost methylation as expected, but some rare cells were able to retain levels of high DNA methylation similar to untreated cells. And because we used index FACS, we also had the cell trace dye on the cells when we collected them. And that meant we could see that the cells that retained high levels of DNA methylation also divided less during treatment. So although plate-based assays really are a pain, there are some things that you can use them for that give you unique insights into the biology. So don't rule them out entirely. So we had information on gene expression, chromatin accessibility, and DNA methylation from this data set. And Sean in my group worked with Kim-An Lecau from Melbourne to use a sparse paired least square statistical method to integrate these different data sets and basically find which genes in the data set were responding to the heterogeneity in the epigenome. And this heat map shows a summary of that analysis with the cells divided into 4 groups across the top and 3 different groups of genes highlighted here. And this dimensional reduction plot shows those cell clusters again. And we first looked at expression of a transposable element, so this is why I asked the question earlier, because these drugs are known to increase expression of transcripts from these repeat elements that are normally silenced. And we saw highest expression of this line 1 element in the cell cluster 3 in aqua. And those cells also had the highest expression of genes that we would expect for HMA treatment. So these drugs are also known to stimulate inflammatory responses, and genes in that gene ontology were enriched in those cells. So it looked like this was a subset of cells that were having the typical response to treatment. However, there was still no difference in DNA methylation averaged across the whole genome in those cells with the transcriptional response to treatment versus the others. So we went on and did even more detailed analysis to look for where methylation in adjacent loci was correlating to expression of a particular gene. And this is challenging, which I'm happy to discuss later. But we also found that this group of genes were enriched for negative correlations between DNA methylation and gene expression. In other words, these genes tended to have lower expression when methylation was in an adjacent locus, and also positive correlations between gene expression and chromatin accessibility. So summing all of that up, it seems like the traditional response to HMA treatment, where loss of methylation occurs at a promoter, for example, and allows re-expression of a gene. This only happens in a subset of cells that have lost methylation and only for a certain group of genes. So there's a lot more that we could be considering that people are usually just conveniently ignoring. Someone just tell me to stop when we run out of time. So the next paper I wanted to show you really draws on what Unica raised earlier about copy number changes. So this is an old paper from 2018, but it was one of my favourites when it came out. And the reason is that they really highlighted the power of DNA methylation data to call copy number variants. This is one colorectal cancer patient, and you can see all the different genetic sublineages in this cancer. They also collected samples from different tissues. So they've got normal control, primary tumour samples, matched lymph nodes, and so on. And then they looked at the overall methylation level within the different genetic sublineages from these different sites in this one patient. And you can see that there's certain genetic sublineages within this tumour that have much less methylation than the other genetic sublineages. So this really allows you to see epigenetic differences in genetic subclones, and there's no other way you can do this. You can't sort genetic subclones as far as I'm aware. So the only way you're going to know about epigenetic differences is if you run some single-cell analysis. So we had a go. This is the sample that Chris and his team are currently preparing for us upstairs. We had to go on this one AML patient that was from Newcastle, from our collaborator. This was a 60-year-old male, and they had a complex karyotype, which included a bunch of different abnormalities. I had to figure out how to read karyotyping annotation when I got this, and I had to teach myself that an isochromosome is when the chromosome breaks in the middle, and then rather than having a short arm and a long arm, you just get 2 short arms or 2 long arms. So this patient had an isochromosome of chromosome 17, and they also had an extra copy of chromosome 12 carrying a deletion and a bunch of other things. So now when you read this karyotyping information, the number in square brackets at the end is actually the number of cells that that karyotype was observed in by the pathologist. So you can also see that there's sublineages here. So the pathologist noted 6 cells with the first karyotype and 4 cells with the second karyotype. So we just sequenced less than 200 cells, but we could already see these different sublineages in our data analysis. So here you can see the isochrome 17, where there's twice as much depth on the long arm of 17 than the short arm. And in this group of cells, cell group 3 here, you can see the extra copy of chromosome 12, but it's missing that little bit. Oh no, got something covering it. Oh, I'll show you later. Actually, it's on the next slide. So here's the chromosome 12 with the gap. So we could group the cells into these different karyotypes, basically, and then we could look in the DNA methylation. So we looked for differentially methylated sites, and this was promoters, and we found that a lot of the karyotypically normal cells that are green here, they actually had less methylation than the abnormal cells. And we also found that the cells I dubbed the major clone, so they made up the majority of the cancer cells, they also had the most heterogeneous DNA methylation. So the cells within that group were more different from each other. So I tend to think of it as the cells are growing really aggressively, they've become the major clone, and they've sort of let themselves go in terms of maintaining their epigenome. This is just my idea, this is not actually proven. But you can see that you can start to ask some interesting questions about how cells regulate their epigenome or maintain their epigenome in the context of a cancer. So that was really interesting, but we also had gene expression analysis on this data set, and actually from that Bjarne paper, from the colorectal cancer paper before. So why am I not telling you anything about gene expression? I'll finish after this slide, or one more. So gene expression can be challenging, especially in primary samples. Bjarne does have a figure on it, but it's not very interesting. It basically just says DNA methylation is low at promoters that are expressed. So that wasn't anything we didn't know. Our challenge was to do with the plate-based assay. So because we were working in plates, then you have so many more batch effects. This is just some quality control data showing that the 2 different plates, red and blue, had very different levels of genes captured in them. So I think this is somewhere where a droplet encapsulation method could really help us. However, I'm a bit concerned about the number of genes that you'll be able to recover, because something else to consider is how many genes do you need to get back to be able to know something about the biology of that cell. Okay. DNA methylation can also work as a natural barcode, so you may not need to do genetic barcoding studies for lineage tracing anymore. Read this really cool paper, and sorry, it's Ken et al. 2025, Nature Methods, and there's been a follow-on up since then. And also, don't forget that single-cell methods can be really useful when you have very rare cells. Not everyone wants to sequence thousands of cells. Some people, like Allegra, have very few cells that they're interested in studying. But that's where plate-based assays and single-cell analysis can reveal really useful or interesting information. In this case, to do with Dunnart or marsupial evolution. Allegra can tell you about that. Okay, so in summary, there are inherent challenges associated with single-cell analysis of DNA methylation, but that means that it's really important to take careful consideration when you're planning experiments, so that you know that your experimental design will give you the information that you're most interested in, and that you can get some unique insights from that data set. Thank you.
Any questions for Heather?
Thanks, Heather. That was very cool. About the lineage tracing, there are some tools out there like NumDat, for example, that are able to call or claim they can call copy number variation and linear tracing from single cell gene expression, and they use that as a matter of course. How have you done that, and how would you compare it to copy number calling from DNA methylation sequencing instead? Do you think one's superior to the other?
I think that when you have a random chance of picking up reads across the whole genome, you've got more power to call copy number changes.
Yeah.
I'm not sure about the resolution of the changes, but yeah, I think that the genome-wide sequencing is just better for copy number variant.
I'd like to make a comment on that too. I really enjoyed your AML thing. Is that a 53 knockout mutant AML?
The first paper?
Stable karyotype AML.
I'd have to check, sorry, the patient data or the cell line.
No, patient data.
Yeah, I don't recall their mutations. I've probably got that written down somewhere. No, they were SRSF.
SRSF2 doesn't usually cause chromosomal. Anyway, so I'm curious about this because we've just done a study on a bunch of 53 mutant and ATM mutant AMLs using Inst. I mean, it wasn't our original goal. It's site-seq sequencing. But the Inst data came out is incredibly. I was really shocked at how good it was actually at predicting predicting copy number variations, and I only found them in the DNA repair mutant AMLs, not in the SRSF2, for instance, or AMLs. And but the nice thing about that was because we had site-seq antibodies on there, which was done with Garvan, we're able to retrospectively gate on those cells what site-seq markers they had on them, and then go and fact sort them. And then show from SMP arrays that they really did have the copy number variations that the first CMV predicted. So I know that, and there were definitely artifacts in the first CMV which we could say that's clearly an artifact because it just kept popping up again and again and again. But the first CMV data was surprisingly good. I'm not arguing that the DNA data would be better.
Yeah, it's interesting, isn't it? Because a lot of genetic evolution studies in cancer just do SNP genotyping.
Yeah.
But you can get quite a lot from structural abnormalities as well, I think. And even I was doing, even looking at immunoglobulin rearrangements, you can see it in the DNA, with the amount of sequencing that we do. You can see if a cell has recombined its immunoglobulin locus.
Yeah,
Yeah,
because the SNP coverage is good enough in those.
yeah. Well, no, just because of the reads.
Yeah.
The random distribution of reads across the genome.
Anyway, very nice.
Any more questions? I would like to thank everyone again for the great talks. If you're interested in running these assays yourselves with us, the SeekGene assays as well as anything from the candidate portfolio, please come and talk to us. We're still here for a bit, so come and mingle. Thank you.